The other thing I want to do is, if you have a question, please feel free to interrupt me. I'm going to try and keep this relatively light and understandable for everyone. Leave some time for questions at the end, and maybe tell you a little bit about where I think science is going at Yachay. So the first thing I'm going to do is get this over here, so I can actually look at you guys. And so feel free to interrupt me. So this is mostly going to be a talk about genomics. And so the first thing I'm going to tell you about is a few things about the human genome. And most of you will know that most of our cells have two copies. One copy is called haploid, and the size of the haploid human genome is about 3 billion base pairs. Now, I don't know whether you are like me, but 3 billion means nothing to me. So the budget deficit of Ecuador is about $3 billion. So just to give you an idea of what we're talking about. That's how many base pairs you have in each one of your cells. Now, the other way to sort of visualize this is to visualize it as if you were to print it out on pieces of paper and put it into books. How many pages do you think you would fill with 3 billion letters? So base pairs are like letters in your DNA, right? So how many pages would you fill with 3 billion letters? Anybody want to guess? 1 page? 50 pages, 10,000 pages.
How many per page?
So you've written term papers, you can get a few thousand characters on a page.
And then 3
billion.
By
It's the budget deficit of Ecuador, basically.
Yes.
So it's about 50 volumes of an encyclopedia, 50 volumes this thick. It fills half of that wall there. That's in each one of your cells. That's how many letters are in your DNA. Okay, most of you will know that there is 23 chromosomes. I'm sure you've all seen pictures of them, and they vary in size. And the thing that I'm going to spend quite a bit of time talking about is something that's called SNPs. Single nucleotide polymorphisms, and they are what make humans different one from another. So we're all human beings, but if you look around the room, we all look different, and so some of these SNPs explain some of the genetic differences between us. And so Dan Larson, the rector, isn't here, but he thought that I was a biochemist, and that's because my PhD is in biochemistry. But the main thing I'm going to be talking about is human genetics and human genomics, and I'll throw in some things in molecular epidemiology and molecular biology. And I think unusually for somebody at this university, I spent the last 35 or 40 years of my life in medical schools. So most of the things I've worked on are diseases. So one of them is galactosemia. We've worked on a variety of cancers: ovarian cancer, prostate cancer, brain cancer, and atherosclerosis, which is not a cancer. But we won't have time to go through all of this, so I'm going to talk about 3 things: galactosemia, prostate cancer, and atherosclerosis. And so, how many of you are thinking about doing a PhD, doctorate? 1, 2, okay. So I'm going to tell you something about how I picked my PhD project, which may not be the best way to do this. And I'm going to tell you how I did this. The first thing you're going to learn is I did this a very long time ago. The next thing is my mentor gave me a project, but at some point I sort of thought that may not work out all that well, so I decided to look for a new project. And I had spent some time in a class called Biochemistry 200, which is at the very top, Biochemistry 202, excuse me, spring of 1983. And how many of you do know anything about glycogen storage diseases? The first 2 lectures in this class. Anybody heard about them? Anybody know anything about them? Good. That wasn't me when I was a PhD student. I couldn't understand it either. So the next thing I couldn't understand was galactosemia, and I'll tell you a little bit about it. So I picked my PhD project because I could understand it. The funny thing about it is, in the same lecture, they also talked about phenylketonuria, which is where I did my postdoc. So let me tell you a little bit about what I did for my PhD, and the first thing you need to do is just say a few things about galactose metabolism. Anybody know where galactose comes from? You know, we all eat food. Galactose is a sugar. Where does galactose come from? You know? Yes. So milk has two sugars in it. One is glucose, which is the sugar that we normally metabolize. The other one is galactose. And so galactose has a special metabolic pathway. And when I was a first-year biochemistry PhD student, I could understand this pathway. And it's a very standard pathway if you know anything about biochemistry. The sugar needs to be activated, and that activation happens in the first step with ATP, which is the energy currency in the cell, to produce galactose 1-phosphate. That then reacts with UDP glucose to form glucose 1-phosphate UDP galactose. UDP galactose can be recycled back to UDP glucose, and glucose 1-phosphate becomes glucose 6-phosphate, and many of you will know glycolysis. That's the standard pathway how glucose is metabolized. And so basically all this pathway here does is it converts galactose into glucose, which is a form of a sugar that the cell can metabolize. And so I told you that galactose, or butyl, that galactose is one of the main sugars of milk. And so there are unfortunately babies who have genetic abnormalities that they can't digest galactose properly. What do you think the outcome is if you are a baby, a small infant that depends on mother's milk and you can't digest galactose? What do you think is going to happen to you? Say it again?
Diarrhea.
Yes, that's one of the symptoms, but it gets worse. And baby poop is smelly to begin with, but it gets worse than diarrhea. What's the main organ in the body that metabolizes most things, including sugars? The liver. Okay, so you're going to have liver problems, and so one of the problems is you're going to be jaundiced, so you're going to have yellow skin. Any other problems the baby's going to have? Turns out that galactose 1-phosphate is toxic, and so you're going to have toxic products floating around, and one of the outcomes of many babies is fulminant E. coli bacterial infections, and so some babies die from this, so it's not a good disease to have. Now there are three different forms of galactosemia. The first one is the blockage of the first step here, galactokinase. The second step is the transferase step, that is the most common form of galactosemia. And the third problem is epimerase deficiency galactosemia. So when I told you how I picked my PhD project, and luckily I did that before the chancellor showed up, and several meetings.
We had to figure out where it was. We were in the other place.
So I just told people how not to pick a PhD project. So I decided to go after the gene that is most commonly deficient in galactosemia. And this is a slide, you know, if you get to be older like me, you can summarize 10 years of your career in one slide. So when I was a graduate student, I set out to clone this gene and managed to do that. I'm not going to tell you how we did that. The only thing I'm going to tell you is something that is very common in genetics, and that is that different populations often have different mutations. And so one of the things that I did as a postdoc then is to characterize these mutations, and one of the things you can see that African Americans have different mutations from Caucasians. And of course, the other fun thing you do in the lab is you genotype yourself and you ask questions, what kind of mutations do I carry? And so I, for example, I carry this variant here that's called N314D that is named after two cities in Southern California, Duarte and Los Angeles. And so, of course, my students decided that that's the explanation why I am as strange as I am, because I carry this variant here. It doesn't cause galactosemia, but it is a relatively common variant. In fact, the allele here that I carry is actually normal in mice, so I'm somehow more closely related to mice than to other people. But I should say this is not uncommon. About 5 to 10% of the population carry this variant. It's also present in monkeys, by the way. When I got my own lab, we decided to clone this gene here, the epimerase gene, and we found the same thing, that different populations carry different variants. Now, from a human evolution point of view, why do you think that different populations have different mutations. So it means that these okay.
Adaptation.
Okay, so one could be an adaptation, and so this is probably something that happened here. So monkeys and mice have the variant, and in people, somehow it became this. The other thing, often are population bottlenecks. As some of you may know, there are diseases, infectious diseases that spread through populations that change the genes. Sometimes populations become relatively small, and variants that were common in that population all of a sudden expand. And the other explanation is after racial and ethnic groups diverge, all of a sudden mutations also arise there because you have very small populations. So this is not uncommon that there are different variants in different populations, and it just tells you something about the history of human populations. The next thing I want to spend some considerable time on is remember we talked about SNPs. These are the things that make each one of us different. And so SNPs, or single nucleotide polymorphisms, occur every 200 to 1,000 base pairs. And remember I told you the human genome is about 3 billion base pairs, 50 volumes, or the deficit of Ecuador. And on the printed page, you can get a few thousand characters. Bless you. I hope it's not something I say. And so SNPs occur every 200 to 1,000 base pairs. So on every single page of the 50 volumes of the encyclopedia we talked about, there will be a typographical error between you and someone else in this room. So human genetic variation is incredibly common. The current reports are something between 20 to 30 million have already been reported. I'll show you some data, time permitting, at the end. But so what we set out to do is to characterize some of these SNPs, differences, genetic differences between people. There are still some pairs up here and here. And then we look at people with and without the disease and see what happens to these people and what do the SNPs do. But we also characterize them functionally to see what the biochemical and pharmacogenetic consequences are. And I'll show you some examples of that. So why should you pay any attention to this? Because you walk the wrong way. This is not where usually the seminars are. So why should this interest you? So the first thing is it may tell you something about genetic susceptibility to disease. I told you that we spend a lot of time looking at a variety of cancers, so it tells you something about the disease progression of the cancer. I'm going to tell you quite a bit about pharmacogenetics. You can think about rational disease prevention, and I'm going to show you an example of that as well. You can engage in pre-symptomatic diagnostics, including in fetuses, if you like. It tells you something about human biology. And believe it or not, at heart I'm also a scientist, so it tells you something about protein structure and function. Now, in the United States, President Nixon in the 1970s engaged in a very expensive war on cancer, so there was a lot of money, including today, that is being thrown at solving cancer. And the question is, did anything come out of this? And if you look at cancer mortality, so cancer deaths in the U.S., from 1975 to roughly 2010, you see that in the mid-1990s there was an inflection point. So cancer kept rising in both men and women, and then it started to decline. So some of this investment in cancer is starting to pay off. Remember, this was started in the mid-1970s, and the changes in mortality we haven't seen until the mid-1990s, so 20 years later. And we're still seeing some of the changes today. And I'll tell you something about cancer in Ecuador in a second too. Now, the cancer that I want to focus on first is prostate cancer. It's sort of an underappreciated cancer in many ways because most guys don't like to talk about things that happen down there, especially when they go wrong. But it's a serious public health problem, and I'll show you some data about that. I spent some time in Australia, that's why that's listed here in the US. But I also show you some actually shocking data here in Ecuador if you don't know what the data is. The disease is characterized by a striking racial ethnic variation and risk, and I'll show you that in a second, and how we looked at that in the laboratory. It's not going to surprise you greatly that risk for the disease is correlated with androgen levels, which are the main male hormones like testosterone. So it's not surprising that it's correlated with those, and we don't know very much about it. Let me show you some general data. Often people know more about, especially in developed countries, about breast cancer than they know about prostate cancer. And like I sort of said, the flippant way of putting it is, women take care of their health, they look after their families, but they also look after themselves. Men don't like to talk about things that might not be working down there. But if you look at the data here for prostate cancer in the highest risk population, which is African American men in the US, is higher than breast cancer in women. The lowest risk population for prostate cancer are Asian men, and it's very low there. I promised you some data from Ecuador. The most recent I could find is from 2008. But the somewhat, to me, shocking, and I'll explain to you in a second why it's shocking, data is that there's 20,000 cases per year here and 13,000 deaths. So more than half of the men who are diagnosed with prostate cancer die from it. And prostate cancer is actually a relatively readily treatable disease because the prostate is a confined organ. So if you excise it before the cancer spreads, you can treat the disease. So the fact that more than half of the men die means that it's diagnosed very late here. Probably most of you know, and I said something about disease progression also, one of the problems with cancer is, do you want me to move? So one of the problems with many cancers, including prostate cancer, is once it spreads beyond the organ of origin, in this case the prostate, it metastasizes in other organs, and once it metastasizes, it's essentially impossible to treat. You can ameliorate the condition a little bit, usually for months, but eventually you'll die for it once it metastasizes. So the fact that more than half of the men die says something about how late it generally is diagnosed here, and so that needs to change. The other thing that's shocking to me, and that's also listed here, in most Western developed countries, but also in many Asian countries, lung cancer is the #1 cause of cancer, and that's because people smoke, and that's highly preventable. In Ecuador, the most common cancer and the most common cause of death is not lung cancer, which is preventable by not smoking. It's prostate cancer. It's more common than breast cancer in women. It's more common than lung cancer. So it's a problem. And like I said, the sort of psychological issue also is men often don't like to talk about things down there that don't work. And so usually the cancer will probably come to the doctor's attention when it's too late, and you die from it. I mentioned to you that androgens play a crucial role, and so androgens have something to do with cell division in the prostate. I'll show you a diagram in a second. Cancer, of course, is uncontrolled cell division, and so it stands to reason that androgens have something to do there. Now, the one thing that's slightly different about androgens from other steroid hormones is that the main circulating male hormone is testosterone, and that is actually not the active hormone itself. The active hormone is called DHT, or dihydrotestosterone, and I'll show you a diagram of that. And what it does is dihydrotestosterone, which is this, binds to something that's called the androgen receptor. They get together and they do a bunch of things, including inducing cell division. And this is a cartoon that shows you that. Testosterone circulates in the blood, way up there. It enters the cell. It is activated by an enzyme that's called steroid 5-alpha reductase. So this thing that I mentioned to you, this compound, is called dihydrotestosterone. And we don't need to worry about any of this mumbo jumbo here. You have dihydrotestosterone, it binds to the androgen receptor, they go to the DNA, they activate a bunch of things. One of the things that comes out of it is growth, and uncontrolled growth is cancer, so it's bad news. And so this is the androgen metabolic pathway, and much like with galactosemia, I decided I was going to look at this 5-alpha reductase gene. And when I say I, it means we, because it means my lab with my students. And so we went after this gene. Turns out that there's 3 different genes. The gene that is most active in the prostate is the steroid 5-alpha reductase type 2 genes. Human geneticists are not very imaginative. It has the acronym of SRD5A2, and we don't need to worry about where it sits. Now, human genes are not contiguous. Many of you might be familiar with bacterial genes, which is a piece of DNA that encodes for a protein. Human genes are split up into these yellow boxes, which are called exons, and the stuff in between are called introns. One of my students sequenced the human DNA from about 100 individuals with and without prostate cancer, and this was to look for SNPs, these genetic differences between individuals. And unsurprisingly, he found a lot. These are all those differences that actually change amino acids, so they might change the function of the protein steroid 5-alpha reductase. And not to worry, I'm not going to tell you about all of them. I'm only going to talk to you about 2, B89L and A49T. And the human nomenclature for these things is the normal amino acid comes first, so A is alanine. 49 is the position of the amino acid in the protein, and T is threonine. So A49T changes a hydrophobic amino acid alanine to an amino acid that has a hydroxyl group threonine. And B89L is valine 89 to leucine. Once we found these things, we asked, what does it do to people? And so we had access to a multi-ethnic population in Los Angeles and in Hawaii, consisting of African Americans, Asian, Caucasian, and Latinos. Remember, African Americans are at very high risk for prostate cancer, Asian are at very low risk. And so in V89L, leucine is the variant allele. And if you look at the frequency of the leucine allele, it's at least 2 times more common in Asian men than as in African American men. So one possible reason why Asian men are at reduced risk of prostate cancer is because about half of them have a variant that may reduce their risk. African American have reduced risk. We also asked whether the variants contribute to prostate cancer itself, and this is data again from this cohort, where we now looked at hundreds if not thousands of samples. And if you look at African American and in Latino men, there is about a 5-fold increased risk for prostate cancer with this particular variant. Are there any questions so far? Okay, I bamboozled all of you, that's good. So people like to talk about a gene for this and a gene for that. So since I just talked about cancer, it's very common to talk about BRCA1. In fact, the acronym already suggests something, that it's a gene for breast cancer. It's not really a gene for breast cancer. It has something to do with cell division. And SRD5A2, the gene that I just talked to you about, is sort of an instructive example of how you don't have a gene for this or a gene for that. You have different flavors of the gene. Talk to you about the variants B89L, where the leucine allele may protect Asian men from getting prostate cancer. And the opposite phenotype occurs if you have the A49 variant, where African American and Latino men are predisposed to the disease. So different variants in the same gene can lead to a relatively different phenotype. So I don't like it when people talk about a gene for this or a gene for that. It's really particular variants that do certain things. And this is an example where we can have essentially opposite phenotypes in the same gene with different spelling variants of the gene. You're ready for some serious biochemistry and pharmacogenetics now? And somehow the chemistry does come in for me. So I'm not going to bore you too much with the details, but I do want to show you a little bit of pharmacogenetics. Now, for the aficionados, the 5-alpha steroid 5-alpha reductase reaction is the reduction of this double bond way over here with NADPH as a cofactor. Turns out that there is a drug that inhibits the enzyme steroid 5-alpha reductase. It's called finasteride. You don't have to be a biochemist to see that there is some similarity between this the structure of this molecule here and testosterone. And finasteride is what people call a competitive inhibitor. What it does, this is finasteride, it binds to the enzyme, it blocks it up, and so testosterone can't enter, the reaction can't occur, so it blocks the enzyme. Now this is not purely theoretical. The drug company Merck manufactures this and sells this under the name of Proscar for the treatment of benign prostatic hypertrophy, which is a disease that older men get, basically they pee a lot, or Propecia for the treatment of male pattern baldness. Other drug companies have other drugs like that. The only one I'm going to show you data on is finasteride, which is the Merck drug. But we've also worked with GlaxoSmithKline on their drug and an Italian company on some other drugs that they have. So drug companies have these things. Now again, I don't want to bore you with all of the details, but I do want to show you a little bit of the data, because first of all, the first column with numbers here is Vmax, which is a biochemical measure for how fast an enzyme works. And remember, I talked to you about A49T and V89L. There are some other enzyme variants listed here. And the only thing I want you to notice is here at the bottom is that there is a 198-fold variation in enzyme activity. And I told you what SNPs do is they make humans different. They make you different from your neighbor. And as you look at your neighbor, the people around you, there are some very strange characters, including the person sitting in front talking to you. And so, of course, you expect a 200-fold variation in enzyme activity. Because we're all different. So why shouldn't there be 200-fold variation in enzyme activity? The other thing that you see there is that similarly for finasterides, there was a 60-fold variation for the efficacy. So the biochemical thing is the inhibition constant, but no need to get into the details. But again, you see a 60-fold variation in how effective the drug is. And what that also shows you, and I'll show you that in another slide more graphically, is what you all know. Drugs work in some people better than in others. Some people have adverse reactions, and some people the drugs don't work at all. Of course you expect a 60-fold variation in the inhibition constant. And so this makes the case dramatically. Blue is for a variety of drugs, the people in which the drug works, and then in, I guess, purple are the people in which the drugs don't work. So doctors prescribe lots of drugs. If you start with the first drug for schizophrenia, and it only works in 1 in 5 people. That's one of the good cases. If you look at some of the others, in many people the drugs don't work, yet doctors prescribe drugs needlessly for people who are taking the drugs with the side effects of drugs and they don't work. So I'm going to show you what we did about that. First thing I need to do though is also introduce you to chemoprevention a little bit, because I'm going to talk about a chemoprevention trial next. Now, most of us, if not all of us, hopefully, brush our teeth every day, and our toothpaste contains fluoride, which in part at least prevents tooth decay. So all of us, most of us, do some kind of chemoprevention every day by brushing our teeth with a toothpaste that contains fluoride. There are some other examples listed here. So if you're a little bit older, like me, you're not unfamiliar, at least with the concept of taking drugs that lower cholesterol, which is the first thing that's listed there. But there are other drugs that are also listed for prevention of diseases. Now, with the data that I showed you, we hooked into something that is called the Prostate Cancer Prevention Trial, or PCPT, to see whether the things we found on finasterides and on steroid phytophylactics have anything to do with prostate cancer and its prevention. Let me tell you a little bit about this trial. It was initiated in the 1990s. For the aficionados, I will tell you it cost $80 million US. Almost 18,000 men were enrolled. They were randomized, and they were either put on placebo, so they basically got a sugar pill, or they were put on finasterides. And they were followed for about 5 years. And again, if you're a little bit older like me, there was a test for prostate cancer that is called this. But in this case, there was a more scientific test for prostate cancer, which was an end-of-study biopsy, and looked whether there was prostate cancer present in these men. The other thing I should show you is that because it enrolled 18,000 men, it did this across lots of places in the United States, essentially all across the United States, and you can see that the major metropolitan areas are represented, because that's where the hospitals and the people are. You may wonder what the outcome of this trial was, especially why older guys like me are not taking Proscar every day. And so the encouraging thing that was found is that taking finasteride for 5 years reduces your risk of prostate cancer by 30%. That's the good news. And Merck, of course, was very happy about this, until you look over here, where you see that there is an increase in high grade, the most aggressive form of the disease. It was very small, but it was statistically significant. And that's why Merck is not selling Proscar, or Propecia, for the actual prevention of prostate cancer. But the big result was, overall, there was a 30% reduction in prostate cancer risk. So, what are the benefits and what are the disadvantages of doing this? So there was a 25% reduction in blood pressure. While we're waiting for that, the National Cancer Institute in the US, which is what funds most of this research, at least in the US, shelled out $100 million for this extravaganza that now is over 20 years old, 25 years old, has started a new initiative, which is an idea where they're going to sequence about 3,000 individuals who have essentially incurable cancers and just sequence their whole genome, the 3 billion base pairs, the budget deficit of Ecuador, the 50 volumes of the encyclopedia, and then use that sequence information to identify drugs that they might respond to that are not part of the general treatment for the disease currently. So this genomic information is now being used at a genomic level to identify new drug targets and try it out in people. So let me stop here again and see if you have any questions about that. Spiros was thinking feverishly. Nope. Okay. So, and the last, because I do want to leave some time for questions. How am I doing? So, I'm not going to bore you with too many details about DHT and activation, except to tell you that we found some interesting issues in genomic databases. All of us nowadays use databases, and I want to show you how sometimes you can't completely trust them. So for reasons that are not going to surprise you, we decided to look at DHT and activation. Remember, that's the active molecule in the prostate that regulates cell division. And we found one variant that we thought might have some activity consequences. In fact, now that you're all familiar with how human mutations are described, this is phenylalanine at position 46. It's changed to tyrosine. And all of us, of course, remember our biochemistry as fondly as I do, so that's the addition of a hydroxyl group. And when you talk to the smart people next door and they do some computer modeling for you of the protein structure, you can see that the additional hydroxyl group here creates a new hydrogen bond, and that actually displaces the NADP as a cofactor, and so all of a sudden that reduction doesn't work anymore. So we thought that that was very cool. We also found that there are lots of SNPs reported in the database, and so this number here is actually the SNP variant in the database, and we thought that they might be erroneous, because if you look at the genomic sequences in the AKR1C2 gene and compare it to the AKR1C1 gene, which people apparently didn't do, that's the difference between these two genes. So we thought that these might be errors in the database. And of course we talked to our friends, the bioinformaticians. I'm not going to walk you through all of this, except to say that I've mentioned to you that there are lots of SNPs now known. You can see there are 61 million already known, and so we whittled that down. Just we started at the University of Southern California, what we think was probably the first molecular epidemiology program in the US and possibly in the world. Ron Ross, who died a few years ago, was my mentor at USC at the University of Southern California. I recall vividly to this day how 2 senior colleagues in biochemistry came into my office and warned me against ruining my career by working with who they call pencil pushers. And so one of them was Ron, the other one was Malcolm Pike. I think we were very lucky in starting this collaboration because we essentially started a new field. And of course, once the grant started to roll in, the people who had warned me about throwing my career away didn't have so much to say anymore and promoted me to associate professor. The other people that are listed here worked on some of the other genes and didn't have time to talk about all of them. Didn't have time to talk to you about atherosclerosis. The one thing I will say about this, you see Jake Luce is listed here. He was the guy I did my first and only sabbatical with. And for those who know anything about Los Angeles, driving from USC, the medical school, to UCLA is like driving to a different country because of the traffic across downtown LA. So I was somewhere else, but we had a great time, and we actually did some interesting things on atherosclerosis as well, and I didn't have time to describe it to you. I've been around to a few places in the world. My lab has been relatively international with people that I've collaborated with. And so I think I'm going to leave it at that and see if you have any further questions. Are there any questions?
Some of the SNPs problematic SNPs were at the end of the exons. Does it mean that it would affect the splicing maybe?
So so that's a very good question. Let me just get out of this and get back to the slide that I think I'm referring to. So didn't talk about this, but I'll just use this slide because it has a lot of introns and exons. So here you can see a ton of exons and introns. And every time we list an amino acid substitution, we list the amino acid substitution. I'm going to try and see if any of these here actually change the splice sites. So here we list the nucleotide substitutions, and some here are at the very end of an exon. So this one might in fact involve the splice sites. We also have what I didn't talk to you much about are deletions, so not just only changes from one nucleotide to another, we also have deletions. Here is another one. So everything you can essentially imagine does happen, including potential changes to the splice sites. Now, anticipating your next question, I'm going to tell you we haven't looked at the messenger RNA and don't know whether it in fact changes the size or shape of the messenger RNA.
The rest of the prostate cancers where there is no obvious connection with the antigens.
Right. So what I didn't tell you is Hodgkins and Hodges got a Nobel Prize in the 1940s for discovering that most prostate cancer can be treated by androgen deprivation, which in those days actually meant castration. Nowadays you would use drugs. But as you correctly say, not all prostate cancer is androgen dependent. And what happens in those prostate cancers is relatively unknown. There is some data that suggests that there is a chromosomal translocation that causes some prostate cancer. So I don't know what you guys know, but often you have chromosomes, sometimes they break and they sort of fuse together in an unorthodox way. Some of these cause cancer. So leukemia is the most common example here of the Philadelphia chromosome, where chromosomal translocation causes cancer. And there are some indications that some prostate cancers may be caused by chromosomal translocation as well, but some may not all. And so to this day, prostate cancer remains a relatively poorly understood disease.
Another question I had, because this has gone to the popular press, what do you think about the potential for diagnosing very early stage cancers without symptoms, practically asymptomatic, by quasi-non-invasive tests, for example, just a droplet of blood or saliva, by targeting specifically microRNAs? Do you believe that this is really something to get excited about?
Well, so first of all, there is a blood test that people use for prostate cancer, which is called the PSA, the prostate-specific antigen. So having blood tests is not uncommon. Now, microRNAs, as Spiros describes, are sort of one of the emerging areas of potential causes of cancer. And there are actually panels that companies sell that have multiple microRNAs on it. And if you have several of them, you might be at increased risk of cancer. I think all of us, whether it's looking at genes, whether it's looking at microRNAs, if we can identify things early, that's better than late. And so when I described to you the data from Ecuador, where there are 20,000 cases a year and 13,000 deaths, one of the real problems behind that is that prostate cancers in this country are diagnosed very, very late. When they're usually spread outside of the prostate, where they've metastasized, and it's essentially impossible to cure it, alleviate it for a few years. So as Spiros says, one of the things that you want to come out of this kind of research are relatively early, often even pre-symptomatic diagnostic tools where you can pick up the disease before it becomes so aggressive that it can't really be treated anymore. So microRNAs are one of the hopeful areas these days. How that's going to pan out, I don't know, but companies sell panels. Any other questions? Yes.
Is the rate similar in more countries like Colombia?
So I have to confess, I have only looked at Ecuador. I don't know, but in developing countries, it is not uncommon that these things are diagnosed. And like I said, with men, there is also the psychological issue. If something doesn't work down there, you don't want to talk about it. And so you only talk about it when it's way too late.
What about those publications that show that they say that an increase in sexual activity is preventing the progress of it?
So while there are reports to that effect, it is unclear that that's true. I guess there are some sort of somewhat more sensationalistic reports that have also made it into the popular press. I am not sure that I believe it, but there are also stories about red meat. Red meat, of course, contains cholesterol, which can be metabolized into steroids, so that, for example, might have some basis. But there are lots of things that happen in stories.
What about gene editing, like gene therapy, local? Is any possible use for that?
So until we know what the true genetic defects are, genetic therapies are probably premature. When I was in Savio's lab, gene therapy, so this was in the early 1990s, was one of the very hopeful things where people were talking about, and so now we're in 2016, and people still talk about gene therapy. People have done it for very few diseases, but so far it hasn't really lived up to its promise, I would say. And in prostate cancer it's even more difficult because we aren't quite sure what the genetic defects are. All right, so next month, Ares Rosakis, probably at the end of the month, and he's the chair or the president of the Comisión Gestora. Thank you for coming.